the session. Actually, I can do that. That's great. Just make sure that's done. Grace, over to you.
Hello, everybody. Thank you so much for coming along to my talk this evening on your Thursday afternoon. My name's Grace. I am a current student at Oxford University doing a Masters of Science in Clinical Embryology, which is a bit of a mouthful, but basically we're looking at early reproductive science and development of the human embryo and kind of translating that into fertility treatments and research within university. I'm with Trinity College and I'm with the Nuffield Department of Women's and Reproductive Health. And today I'm talking about molecular biology with a quick recap of hopefully some familiar topics that you would have covered in biology. And if it's new to you, hopefully you can get some new knowledge. And then we're going to have a look at the genetic code, the Human Genome Project, and finally looking at how this all applies to reproductive science and what the picture is looking like today. Thank you. Next slide, Richard. So a little bit about me before I go on to the kind of the substance of the talk. So as you may have picked up, I'm not UK born and bred. I'm from New Zealand, which is pretty much as far away from England as you can get. It's on the other side of the world. And I went to University of Otago, which is down the bottom there in a small town about the same size of Oxford, actually, called Dunedin. And that's our beautiful clock tower there. There's kind of a nice kind of area in the middle of town. Like Oxford, it was quite cold, so it didn't always look that beautiful, but it was a really wonderful place to go to university. And I picked Dunedin because I wanted to go to medical school right after university. And there's two medical schools in New Zealand, one in Auckland up the top of the country and one at the bottom. And I chose to move away from home, so it was an easy choice to go to Dunedin. Next slide, please. Thank you. So my plan on leaving high school was that I was going to enter the foundation year of university, which is kind of a common pathway. And if you do well in that year, then you can be admitted into medical school. And then I wanted to be a doctor after that. So I'd really enjoyed biology at school, loved science. I knew I wanted to do something medical, and this was my plan. But my first year of university, there was a lot going on. You move away from home, you meet 100, 200, 300 new people. Suddenly you've got friends, social plans, and there's a lot going on. And so I worked really, really hard in my first year of university, but it didn't quite go to plan, and I didn't get the acceptance into medical school that I really wanted. So then I had to stop and think, and I thought, right, well, I really wanted to go to medical school, but what are my options now? And I was really fortunate in that I had really loved a lot of what I had studied in that first foundation year of science. We covered a lot of health sciences, a lot about human biology. And I thought, well, I'm going to use this foundation year and put this toward a science degree. And so I went on then to complete a science degree in genetics and worked really, really hard in that degree and managed to then get into medical school via the postgraduate entry. Next slide, please. Thank you. So while my plan A didn't work out, my plan B was eventually I did go through medical school, and I've since been working as a doctor in New Zealand for 3 years. And I think I just wanted to convey that at the time I was really upset that I didn't get into medical school. It seemed like a lot of my friends who had got in were far ahead of me, but there was a real door opening for me in that I managed to do a degree in genetics that I loved. It's completely shaped where I'm taking my career now. There is so much common ground between science and medicine. And so I think if any of your plan A's don't work out, whether that's medical school, law school, or even your university or town of choice that you're thinking about studying in, there's always more opportunities to come. And I'm really grateful for the journey that I've taken because it's taught me things that I don't think I would have had the choice to otherwise. All right, so this is me here in Oxford. So I made it this far away around the world. That's our famous Radcam in the middle of town. So that's our stunning library where I try and study when I'm feeling motivated. And that's me on the right in our teaching laboratory at our science centre. So what you're seeing there is me with this very fancy, almost like robot type microscope. And on the right-hand side of the screen, you can see there's kind of two needles that are holding this little blob in the middle, and that blob is an embryo. And what we're learning how to do is learning how to biopsy the embryo. It's an animal embryo. We use them for training with a view to test that embryo for certain genetic diseases. And that is what they're doing in the laboratory at the moment for fertility treatments for patients. So that's me being very happy on a good day in the lab. All right, so that's enough about me. Let's go on to the talk about the proper science, which I'm sure you're all really interested in too. So molecular biology is kind of the topic of my talk, and what molecular biology means is kind of the really fundamental physiology and really chemical makeup of a cell. So you've got your cell there, which I'm sure you're all familiar with, and at the very heart of the cell you've got DNA, which stands for deoxyribonucleic acid. This is packaged really tightly inside the nucleus, which is the red blob right at the center of that cell there. And every single living organism has it, from plants to bacteria to viruses to humans to animals. Everything comes down to DNA. And at the very center of the cell, it really is the core, because it provides all the instructions that a cell needs to function, to communicate with other cells. And so it gives kind of the instruction manual to life. And so I think of, the DNA as kind of like a recipe book, and it has all of these recipes for different parts of the cell, different proteins, and through the cell making different recipes, it can help go about its function. Next slide. So who discovered DNA? So DNA has this very typical kind of ladder appearance on the right-hand side. So if you picture a twisted ladder, that's what it looks like. And this was very famously discovered by two scientists in America, in New York, called Watson and Crick. And they're doing a bit of a funny pose there. I think if this was Instagram or something today, they would be posing for the camera. But there's Watson on the left and Crick on the right, and they're looking quite happy because they've cracked the structure of DNA. And if you use a bit of your imagination, you can see this kind of assembled structure behind them looks a bit like a twist, and then you can see these are the chemical structure, and the twist there is really similar to the twist that you see in the ladder on the right. And that was in 1953, so a long time ago, and they went on to win the Nobel Prize a few years after than that. So that was a huge foundation in the history of molecular biology. But there's one other person that needs to be mentioned here, and we can't go past kind of mentioning her. So if you click for the next animation, Richard, yes, perfect. So that's Rosalind Franklin. So she, being a female in the 50s, was outnumbered vastly in her lab by the men. But she had all of the brains that men did. She had a PhD at Cambridge, and it was her X-rays and her techniques and her equipment that ultimately enabled Watson and Crick to put the information together to figure out the structure. So she developed the imaging and this X-ray crystallography, which was hugely advanced for its time, to look at this DNA structure under a microscope. But sadly, she died in her late 30s from cancer, which was probably due to the radiation that she had used to develop these techniques. And she passed away before she could be awarded a Nobel Prize. So whenever you think of Watson and Crick and their Nobel Prize, think of Franklin as well, because she was equally deserving of that prize. And many people think that if we rewrote the textbooks in history, we should be putting a Nobel Prize next to her name as well. So back to DNA. So Watson and Crick and Franklin had found this DNA structure, and they knew that it was this double helix, so this twisted ladder. And we also found out that DNA is not one continuous long strand. So you can see the strand on the left there, the twisted ladder, is actually broken up into smaller chunks. And these chunks are called chromosomes, and they have this very typical kind of X-shaped kind of appearance, and they look like the letter X. So the entire human DNA is chopped up into these small segments, and they're organized into pairs because you get one half from your mum and one half from your dad. And they're packaged into the nucleus where they're kind of floating in space. And if we lined them all up in a picture, they would look something like you can see below. So humans have 46 chromosomes, that's 23 pairs. 22 of these are called autosomes, and they contain material for functioning in cells and tissues in the body. And then there's one pair of sex chromosomes, which determines whether you're biologically female or biologically male. So you can see 1 to 22 laid out there, and then the X in the bottom right-hand corner, there's 2 X's there. So this is a female person. So that's how DNA is organized within the cell. Perfect, thank you. Next slide. And so then they realized, well, within all of these different chromosomes, surely these different chromosomes do different things. And so the next step was to figure out exactly how this ladder and how this appearance of DNA was correlating to information and kind of instructions for the cell. So if we take this DNA ladder on the left and we untwist it a bit and lay it flat, you can get this appearance in the middle in that picture there, where you get this sugar phosphate backbone. So that's like the tall bits of the ladder that go up and down. And then you get the rungs that go in between. And these rungs are called base pairs or nucleotides. And you can see they've been given different letters. So cytosine is in purple, and that always pairs with a G, with guanine. And then below that is an A and a T, adenine and tyrosine. And so as this DNA kind of runs along the ladder, and this ladder is very, very, very long, and if you think of a ladder with all of these rungs, then suddenly you can start to see that you get these repeating letters. And if you read the letters along from top to bottom, so let's say we start at the top left, and we go C, A, C, G, A, C, T, T, and we write that out as if we're reading a book. The letters aren't the same, but the idea is similar. Then suddenly on the right-hand side, you can see you can start writing these letters out. And that all looks a bit like nothing, doesn't it? You can't even make words out of what's been saying there. So it looks a bit like, same 4 letters repeated over and over and over and over again. But the ordering of those letters is really important, and that's what is called the genetic sequence, so the sequence of the letters. And so scientists began to realize that it was the ordering of these letters within DNA that was giving them some sort of code. It was telling them something. And they needed to find out, what is being told, what's the story that's being told in these letters. And they realized that if they can put all of these letters together, then perhaps they'd be able to find out, how does a cell turn this into life, really, into a functioning cell. So if we took the whole genetic sequence, that's called the genome. So the entire sequence from start to finish is called the genome, all of the letters lined up. So next slide, please. So in the early 1990s, as scientists get more information about, the letters and they started to realize that, hang on, we need to sequence all of these letters. We need to figure out how these letters all fit together. And this was over 20 years ago now that they completed it in 2001. It took 13 years for scientists to get enough information, enough technology. And they were really, people talk about kind of the greatest space race as the greatest event of our generation. But I think this here is probably the greatest, perhaps the greatest next achievement after the space race. And that it took 13 years to do. It cost over $1 billion for them to find the order of these letters. And that's because they really had to invent these technologies alongside the, that they didn't have the technology to do it. They had to stop and think, we've got to invent the technology to find these sequences. And many labs around the world with hundreds and thousands of scientists all pulled together their data to kind of compile this first draft. So along this graph here, you can see 2001. If we go up on the x-axis in the bottom, 2001, if we shoot to the top of the y-axis, we can see it cost $100 million, so $1 billion. So that was 20 years ago. So really interestingly, since then, you can see the cost of the genome drop, and then suddenly it just falls at about 2008. So nowadays, if you extended this graph out, the cost of the genome would probably cost about $1,000. So, £800 to £1,000 or something to sequence the human genome. So there's been this rapid kind of explosion of technology, which has allowed the genome to be done cheaper, faster, more accurately. Every single day there's new technology coming out on the market to enable scientists to get through all this data and information and do it. And one example of this is that now it's even become commercialized. So the sequencing of the DNA is not something that's just happening in labs for research purposes. It's actually being made available to the public. So you've got a company called 23andMe, which put together this kind of very snazzy looking box that can arrive in your post office. And you can order these on Amazon for, I think it's a few £100s. So, it's not cheap. But if you look back to $1 billion, it's pretty phenomenal that 23andMe and 23 because there's 23 pairs of chromosomes. And they can they don't sequence your whole genome, but they sequence a few hundred parts of it that tell you interesting things. So it might tell you, maybe you've got a common ancestor back to, early Africa, or maybe you have a small, inherited part of your DNA that has connected you to somebody else in America, or it might tell you a bit about disease kind of susceptibility. So we've come so far from 2001, where it took 13 years and so much manpower and woman power to do this, and suddenly now we've got Amazon delivering a kit in our mailbox the next day. So this is an incredibly exciting field and a really wonderful time to, if this trend continues in your scientific careers, there's a lot of opportunity for some really impact here. Next slide, please. So let's have a think now, and I'm going to open the floor to Slido questions. So when they put together this first sequence of the human genome after 13 long years, and they lined up all of the letters, how many letters long do you think it was? So now I'm going to flick to Slido, and there should be a multiple choice question there for you. And I'd like you to have a guess at how many letters long you think the human genome was.
I'm not wanting to build up the suspense, but I'm just going to pause a moment before showing how people are voting, just so you're voting sort of independently rather than just
have a think. There's
seeing
no,
how other
it's all
people are voting.
anonymous. There's no bad answers. This is a learning thing. Amazing.
Can you see that, Grace? Yeah.
Yeah, I can see that. So yeah, about 2/3 of you have said 3 billion letters long, which is absolutely correct. It's actually 3.2 billion, but I gave you 3 billion, and I think you've done really, really well. And 3 million letters, I agree with you. 3 million letters is still a lot. So, and 303,000 are incorrect. So 3 billion letters long. So this was phenomenal because they suddenly had they had got the code in front of them. But then the next step for them after that was to say, well, what do we actually do with 3 billion letters? So if you go to the next slide, please. So to get an idea of 3 billion letters, so that's 262,000 pages or 175 large books. And I would say I'm not a very fast reader, but and some of my friends are fast readers, and even my friends who are fast readers, I think, would take many, many years to get through 175 large books, let alone even understand what had gone on. And remember, it's just the same 4 letters over and over and over again, so it doesn't read like a book. There's no instant message there. So they realized that, right, there must be some parts of this sequence of this genome that are more important than others. We have to go looking for the good stuff. And I like to think of this as a recipe book. Imagine you've got a recipe book and you've got a few blank pages and a few picture pages, but what you really want to get to is you want to get to the recipes, particularly the brownie recipe, because I think that's my favorite recipe. But you want to keep looking for the recipes. And if we think of the next step then was for them to look for recipes, and that is in the form of genes. So if we go to the next slide. So what is a gene? So a gene is a DNA sequence, like a recipe, that is in a very defined, discrete region of the DNA. So it's a section of DNA that provides very explicit and very detailed instructions to the cell. And it does this by giving information to make proteins. So on the right-hand side there, you can see that this stretch of gene there is giving information to make proteins. And proteins are used by the cell for so many different things. So they might be enzymes, they might be structural, they might be support, they might be channels. And so it gives the cell its identity, its combination of proteins. And this means that, depending on what genes are used and what proteins there are, it means that you can use the same DNA in the same cell, because remember, the DNA is the same in every cell. You can use different recipes or different genes to make different proteins, and that ultimately gives you your different cell types. So a brain knows that it's a brain, a liver knows that it's a liver, a brain cell knows that it's a brain cell, all because of the different combinations of genes that are active in that cell. Next slide. So the next step after this Human Genome Project was to say, we've got this sequence, let's see how many genes we can identify, how many recipes can we find. So if we took Beyonce's genome and we put it through one of these fancy laboratories, or not even fancy, it would probably only take a day now. Remember, 20 years ago, it took $1 billion. She'd probably kindly pay for it because I think she'd be able to afford that. But say she gave us funding and she said, I need you to sequence my human genome. How many genes do you think we would find? And so if we go back to Slido, I'd like you to have a guess at the multiple choice and see if you can have a think about how many genes we would find within those 3 billion base pairs. Great, I can see some answers coming in. Thank you for your participation. We've got a few options for all of them. Keep guessing, this is really good. All right, excellent. So 191,000 is by far and away the most popular guess, followed by 22,000, followed by a few for 5,500, and then no takers for 400. So very interestingly, the answer is 22,000. So many of you, rightly so, thought, let's go for the biggest number because that's what I did before. But I wanted to fool you a bit, and it's really interesting because when the scientists sat down with the 3 billion pairs, they also thought that they were going to find about 100,000 genes. And it was to their huge surprise that they found only 22,000. And they were kind of a bit disappointed by this because they thought, humans are really intelligent, compassionate, sophisticated, essentially animals, but, creatures, surely we have the most genes, but that wasn't the case. So if we go to the next slide. So 22,000 genes is a lot. So how does this number compare to other living things? This little flea of an insect down here is the animal that has the most genes, and that's 31,000. This pine tree here has 91,000 genes. So, a simple looking tree has, almost 3.5 times the number of genes that we do. And then on the right-hand side, that's coronavirus. And coronavirus only has 15 genes. And that is phenomenal, thinking about the impact of a virus built on only 15 genes. So, as all of these other genetic sequences were being discovered, scientists were, they thought that they had cracked the code to life. We'll have 200,000 genes and we'll be superior, and that's that. But, they had a problem on their hands. They had to explain how only 22,000 genes led to, humans being the way they are versus, for example, a pine tree. And they found that, as I said before, it's a combination and how these genes are kind of used and everything that is a marker of sophistication. And the picture is a lot more complicated than we originally thought. Next slide. So what have we learned? So we've learned that we're actually really similar to other species. So alongside our genome being sequenced, they were sequencing every animal sequence has had their genome sequenced. So 95% of our genome, all of those letters, we share with apes, and 50% we share with bananas. So next time you're having a bad day and you don't want to do your homework, you can say, it's probably because my banana side of me is coming out today and I don't really feel like doing my work. And so genome size, they realized, and the number of genes doesn't necessarily correlate to biological complexity. And in fact, there's other things within the DNA, the non-genetic regions, that are starting to become more and more and more important in how a cell functions. Next slide, please. So since the Human Genome Project, we've managed to sequence, thousands and thousands and thousands of different people's DNA. And we've learnt about mutation. So this is when typos are in the sequence. So if you get a wrong letter in the wrong place, you can have problems like cancer, diabetes, and disease. And so there's a lot of genetic diseases have been discovered. We can also come up with new medication targets. So medications and tablets that enter our bloodstream and into our cells can target areas of the DNA based on their sequence. We've also come to understand how the environment acts on our genome. So different genes can be turned on or turned off, or active and inactive, depending on an impact of environment. We also have learnt about inheritance. So a lot of these conditions, remember, we pass our DNA from one generation to another. So a lot of inherited conditions are passed through DNA. And so we can learn more about inheritance patterns and also testing for these conditions. We've learned a lot about forensics. So you hear about DNA testing on, crime scenes in the movies, and hear about, they've managed to implicate someone in a crime because of their DNA. And that's all thanks to the Human Genome Project. And then beyond humans, we've also, as we said before, sequenced different plants and different animals. So agriculture and livestock. Now we can selectively breed different species of animals so that we can, have better milk production, or we can grow more crops. And so we've really learned so much in this technology boom from the Human Genome Project that's been applied beyond the scope of human medicine. Next slide, please. So the final part of my talk kind of applies this to my field that I'm particularly interested in. So developmental biology, or reproductive biology, is the study of how a sperm and an egg, so on the left-hand side there, you've got a sperm entering into. It's not actually an egg. An egg is a lot larger than that. I think they've just done a nice little bubble for some from Photoshop or something, but that's what it looks like in essence. So remember, you get half your chromosomes from the sperm, and half of them are already in the egg. So from that moment onward, how does that fertilized egg become a human? And so every single thing that you need to become a human is contained within that fertilized egg, which I think is just the most phenomenal thing. So a fertilized egg eventually goes through the developmental stages in these gray boxes here. So on day 1, you can see that those 2 little blobs on the inside are the 2 nuclei of the cell. So remember, one has DNA from one has DNA from the mum, and they fuse. And then on the next slide in the middle at the top, and the next picture in the middle on the top, you can see now that looks like there's 2 cells there. So it looks like the top half and the bottom half have now separated. And then by day 2, so only 24 hours after that egg has been fertilized, you've now got 4 cells. So you can see 1, 2, 3, 4. And then by day 3, if you use your imagination, you can see 8 cells. And by day 4, you've now got what is a clump of 16 cells. And then by day 5, you've got, that's an early embryo there. So that's only 5 days old. It's still very much a cluster of cells. It doesn't look anything like a human or anything like that. But it's the very early stages of all of those cells there saying, right, I'm going to become a brain cell, and I'm going to become a liver cell. And so this day 5 little cluster of cells is suddenly organizing itself into these kind of primitive or early tissues that will eventually become different organs, your brain, your eyes, your ears, your left hand, your right hand. It's all, there's millions and millions of genes that are being turned on, and these proteins that are telling the cells around it what's happening. And then ultimately a pregnancy takes 9 months, and then a little baby comes out at the end. So this is really, I think, the beauty of genetics, that everything that was needed, all the instructions that we ever needed were inside that original fertilized egg. And then 9 months later, a human comes. So how does a cell know what genetic sequence to use? And the answer is this is what we're all still trying to solve. The picture is incredibly complicated. If you pictured every last, chemical, every last protein in these cells, you'd have, you couldn't read them all off on a list. There's so many different pathways. And then, how does a brain cell know that it's a brain cell? Next, the cell on one half of that little cluster might be doing one thing, and then on the other side, it's going to be something completely different, maybe a liver or a lung. And so early developmental science is trying to figure out how these pathways and how these cells are committed to becoming the organs that they are. And I think this is really, really, really interesting. There's a huge amount of research going on about this, and we're learning about really how life begins. Life begins at the very first few days. Next slide, please. So the final part I want to talk about is genetic editing. So we've talked a lot about how genes go on to make proteins, and it's the proteins that give the identity to a cell. And so once this human genome was completed and they had sequence in front of them, it became very obvious that some parts of the cell were responsible for certain things. And we talked before, about what we've learned from the human genome. And we know that, for example, some parts of the human's genetic sequence can have typos, and that can lead to disease. And then other parts can be turned on or activated, and other parts turned off. And very famously, a lot of you who have been keeping up with science news may have heard about CRISPR-Cas9. CRISPR is an acronym, and it refers to basically a cut and paste technology. So CRISPR is an enzyme, and there's a picture of it here on the left. And if you look at that green blob with kind of all the pokey bits, you can see that that's the green protein there. And it's almost like as if a hand is reaching out toward you. It's holding a piece of DNA. So the purple and the blue is a DNA. And what it's doing there is that it is cutting the DNA. So you can see that the strands of the DNA have kind of been pulled apart a little bit inside that palm of that hand. And what it's doing is that with that new yellow piece of DNA, it's inserting a new piece of DNA into that genetic sequence. So it's rewriting, it's cutting and pasting a part of a genetic sequence. And this technology here is the first kind of well, not the first, but probably the most famous technology we have for rewriting the human genome or any genetic sequence. And you can see this has massive, massive potential. So we talked about what we've learned before about the genetic sequences. And if we look at the opportunities for genetic editing, so we can have targeted medicines. So we could give some DNA to somebody and say, right, you're going to perfectly respond to this medicine that we're going to give you, because this DNA will give you the capability to do that. We could make new biofuels. So we could construct new plants or new different, maybe bacteria that produce different types of oils for cars. We could make disease-resistant crops. So let's say you've got a corn crop and it has a really bad fungus infection that wipes out entire crops. If you gave it a gene or a bit of DNA that made it resistant to that fungus, then suddenly you've made a crop that is not going to have any problems with disease. And so think of the impact on farmers. You could have whole livelihoods destroyed by crops in a bad season, but this wouldn't be a problem anymore with genetic editing. We could make new industrial products. So we could make new products for labs, new resources for new science innovations. So it's really, really exciting opportunity. Technology will change our lifetime. It's still in its very early days, but these areas are all being researched and are all likely to happen in some shape or form within our lifetime. But as a lot of you are thinking, gosh, it's not all good news. Surely there are some problems with this. It seems really risky, doesn't it? So the first and utmost there is safety. So what happens if something goes wrong? What happens if, do you mind going back? Yeah. So safety there. So what happens if something goes wrong? What if CRISPR enzyme, the green enzyme, gets it a bit wrong and puts the DNA in the wrong space? What if you want to change your eye color from blue to green, but suddenly that blue DNA ends up somewhere else? It's got the potential to, cure disease, but you don't know what if it goes wrong. You have to be prepared to safeguard against that. And really concerningly, this brings up a lot of ethical concerns. If we're chopping and changing someone's DNA, are we allowed to do that? Would you do that to your unborn child? Would you do that to your grandmother? Would you, and you could say, well, maybe I'll just make my child's eyes blue, instead of brown, because I've always wanted a blue-eyed child. And, suddenly, blue-eyed babies become trendy. Do you want them? And what happens if it goes wrong? And so the regulatory challenges are here. So there's huge committees of ethicists, scientists, politicians, science communicators who are sitting down and saying, well, the future is here. CRISPR is here to stay. There's, thousands and thousands of labs around the world that are researching CRISPR on humans, on plants, on bacteria. How do we regulate this so that this technology doesn't get out of hand? Because in the wrong hands, this technology could potentially be really dangerous. Firstly, we don't know if it's safe yet in humans. There's been some very early trials, but it's very, very specialized, and labs need licenses to use this. So we don't know fully yet if it's safe in humans. And even if it is safe, should we be allowing this? Should we be allowing genes to be cut and pasted and changing the makeup of our very being at the core of our cells? So it's a very difficult question, and that's one of the reasons why I love this field so much, is because the technology is moving so quickly that the regulation and the ethicists are, it's an arms race. Everyone's trying to keep up with each other. And we have to, as a scientific community, allow the regulations to be lenient enough to give opportunities for new research, but we have to make it safe for, potentially for clinical trials, for scientific trials, before we offer a commercial product. Because, there have been a lot of historical shortcomings with technology that's been let out too soon, basically, and people have been harmed from it. So there's a real obligation and a real ethical issue here in making sure that this technology is safe before it goes out into the big open world. So next slide, please. So with that very brief summary on genetic editing, you've probably, I'm sure, hopefully it's kind of made you ask some questions. I would love for you now to have a think about, maybe there's one or two adjectives that you think that you're thinking right now about genetic editing. Are you excited? Does this sound like the best thing ever? Are you scared? Do you think that this technology is going to do more harm than good? Maybe you're really bored and you don't want to listen to my talk anymore, or this is something that is not of interest, and that's okay too. But if you're feeling something, and often this does kind of invoke kind of really strong emotions in people because I think it's really challenging. If you'd like to write something on Slido, I'd ask for the Slido to be pulled up right now, and let's see how you're all feeling about this. Wonderful. We've got some really good answers coming in. Fascinated, yes. Excited, hopeful, really good. Anxious, I completely agree. This technology is really nerve-wracking. We're pushing the boundaries of science like it's never been pushed before, and the ball is rolling, it's not going to stop, and so it's really important that good quality research comes along. Incredible, yes, I agree, incredible. Where does it stop? I completely agree. There's that slippery slope argument of how far do we push this? Good for genetic diseases. Yes, I think we'll be able to cure some genetic diseases in our lifetime. Risky for designer babies. Yeah, exactly. Fantastic. So you've really covered so many good aspects that I completely agree with all of those things there. I think really similarly along all of those lines. I do like that, a lot of the central ones and the biggest ones are positive and optimistic. So hopefully amongst all of you there, we've got some future scientists that are potentially, taking the lead on this and saying, right, well, maybe I'll be the one to steer in a really revolutionary experiment in CRISPR. Maybe I'll be the one to sit on an ethics panel or communicate. Science communication is really important, communicating to the public. So I'm really pleased that you find this as interesting as I do. Next slide, please. All right, so we've touched a little bit on this so far. So can we use CRISPR to change the human genome? So in theory, yes, we can. So at the moment, it's currently illegal to do it on humans. There was a really famous case 2 years ago in China where a scientist did it on humans and he ended up in prison because the entire scientific community said CRISPR is not yet ready for human use. So that was a really important event 2 years ago. It shouldn't have happened in the first place, but it can tell you a bit about what the scientific community thinks about CRISPR on humans at this stage. So I think the theory is that it's not yet, but at some stage it might be possible. So designer babies, people think of designer babies, and I mentioned before about, picking eye color and picking, freckles or not freckles or height and things like that. And I've simplified it a bit. There isn't one single gene for any of those factors. So it's not going to be a case of picking a single gene to make your baby intelligent or funny or left-handed or right-handed. A lot of what makes us human doesn't come down to one single gene. And so designer babies, as it stands at the moment, is very much a science fiction construct. But I think all the elements that we've come to discuss and have thought about before makes you think that in the future, a long way in the future, maybe it will be closer to reality than we think, and we have to be prepared for that scenario. Next slide, please. So that concludes my presentation, but I've got one last slide here as a plug for women in science, technology, engineering, and mathematics in STEM. So as a woman in science, I think it's really important to encourage other women to get involved in a field that has really historically been very male-dominated. And at the beginning of my talk, I mentioned Watson and Crick, and then Franklin not getting Rosalind Franklin not getting the credit she so very much deserved. So I think we've come a long way. 2/3 of my med school class was female, and the majority of my science MSc class, and now that I think about it, also my bachelor's, my genetics class, are female. So certainly in my classrooms, I haven't been made to feel like I'm not supposed to be there. I have always felt that my voice has been heard, but I know it hasn't always been the case. So I think if we can encourage more women to get into the science and normalize women in science, then we're going to have really great representation. And it's really important that if you are thinking about it, you can do it. So if you want to do science at university, I would say go for it. It changed my life. And I'd like to acknowledge Jennifer Doudna and Emmanuelle Charpentier here on the right-hand side, two stunning female scientists who won the Nobel Prize last year for their work with CRISPR. And I was lucky enough to hear Jennifer Doudna talk in a YouTube talk a couple of months ago for our university, and she was seriously impressive. So two inspirations for you. If you have a moment to look them up, I would encourage you to read a bit about what they're studying and how they got to where they were. You never know what you're going to be asked in an admissions interview, and this could be a really good opportunity to kind of show that you're up to speed with what's happening in the scientific field with some reading about them. Yes, so next slide, please. Oh yes, and I've just put little crowns on them because I think they're absolute queens. There we go. All right, so that's my talk for today. I've gone on quite long. Thank you so much for listening. That final slide there is a picture of a zebrafish embryo with some very cool fluorescent stained genes. So the head is at the top and then the spine kind of curves around to the right with the tail in red. So science can be very technical, but it also can be very beautiful. So that's a bit of what we're looking at in the lab at the moment. So I'll conclude there. Please do write any questions that you might have on Slido. No question is a silly question, so I hope I can help you if you've got anything you'd like to just discuss further. And thank you so much for your interaction and for listening. It's been a real pleasure. Thank you.
Thank you so much, Grace. If as a participant you were here after we were talking about how to access Slido, if you go to sli.do or just Google Slido, it'll ask you for an event code, and tonight's event code is Trinity Talks 8. Trinity Talks 8, with both the T's, the first T's of each word in capitals. Okay, we've had some great questions. I'd just like to start with a statement that's on Slido, which is quite unusual. This is incredible. I'm seriously considering a career in this field. So thank you for that positivity there. We'll start with some questions relating to some of the specific content. So kind of go specific. There was another comment as well, and that was one of the comments that was some of the comments went to the word cloud, but also how you're feeling about the sequencing of the human genome. Excited, but I think it needs to be regulated in the correct way and only used by people who know what they were doing. So how can a virus such as the coronavirus have so many mutations, even though it doesn't have that many genes?
Yes, such a good question. So remember that every single time a cell divides, so all of our cells are very active, they have a certain lifespan, and our cells are all dividing in our body over and over and over again. We've got billions of cells in our body. Every time a cell divides, it needs to make a copy of the genome so that when the cell splits, there's equal number of chromosomes left in each cell. And it does this by, there's enzymes in certain machinery that makes a copy of the DNA. And humans have really, really good proofreading machinery. So we can go back and proofread along the sequence and make sure the letters are all in the right order. But the way in which viruses replicate their genome, they don't have very good proofreading machinery. So they prioritize speed and replication and just sheer volume of numbers. So they just say, right, we're going to make as many copies as possible, and we don't really mind if they have mutations. And so therefore you can get bad proofreading and you can get typos in the sequence. Sometimes those typos won't mean anything. They're in an area of the genome that doesn't do anything. Sometimes they might mean that the virus can't survive at all, so it's a fatal mutation. And then other times the mutation might give the virus a new property. And so, to bring it topically to coronavirus, you've probably heard a lot about spike proteins and what's happening on the surface of these viruses, and that's a lot of what the vaccines are targeting. And so in a pandemic like this, if you think about how many viruses are out there and how many times it's replicating, the potential for these errors to come in is really quite apparent. And so viruses without their proofreading machinery can make mutations fairly quickly. And sometimes those mutations might give a new advantage to the virus. And so that's what we're watching and hoping for at the moment with, containment with lockdown versus vaccines and social distancing, is all trying to stop this replication pattern. Yeah, really, really good question. Thank you.
A similar question in a way, how come a tree has more genes than a human, even though humans are a more complex species?
Yeah, great question as well. So that was the real surprise after the Human Genome Project. So what they've come to realize is that initially they thought that number of genes was the kind of the hallmark of complexity, and number of genes correlated to complexity. But what's really interesting, and I could talk forever about this, is that the total genes inside the genome only makes up about 2% of all of those letters. So there's 98% of letters that have some other function. And what we know now is that it's not just the genes and the proteins. It's sequences or stretches of DNA that don't look like genes but are behaving similarly or behaving a little bit differently. And so initially they thought that 98% of the genome was all junk, and it was just had just stuck around through many years of replication and everything like that. But now they've come to realize that there are actually little recipes in those junk bits of DNA that aren't junk at all, that are really functional. And so in the last 15 years, there's been a real search and a hunt for maybe, a new type of gene or a new classification of a gene that we just don't know about yet. And so I think as time goes on, we'll find out that humans, what we thought was mainly junk in humans, will actually be all functional material that just hasn't been called a gene in the way that we thought genes looked like 20 years ago.
Wow, that's mind-boggling. A couple of questions about CRISP. Is that how you pronounce it, by the way?
CRISPR, like
right,
potato crisps. Yeah.
Okay, one that's more broad. Firstly, how do you envisage the field you're currently working in being affected by CRISPR?
Yeah, so I work in fertility, and that picture of me in the lab with the embryo. So, the really obvious application for CRISPR would be if you took the fertilized egg. So the minute the egg had been fertilized, before the cell had divided, if you were able to cut and paste DNA at that point, then every single cell after that, so every single cell that went on to become a human would carry that cut and paste segment. And so if you were able to cut out a disease, and then you could grow a baby, you could transfer that embryo back into a mother's womb, and you could grow a baby without a disease, then you've essentially cured a disease at a very core level, which is pretty phenomenal.
Wow. And I suppose a related question, you've already partly approached it. When do you believe CRISPR will be tested on humans?
Yeah, so at the moment it is being tested on humans, but it's highly, highly, highly regulated. So only a handful of labs around the world are able to do it on fertilized eggs, and no babies are being born with CRISPR at the moment. Not even close to that. So I think, really $1 million question. I think there's a lot more research that needs to be done. There's some early trials that have shown it's really non-specific, so it's copying and cutting and pasting where it shouldn't be. And so we need to create better lab conditions and more specific kind of tools for it to do its job properly. But I would say, potentially within a decade, we'll have to come back and reconvene in 10 years and see if I'm right. But I would say probably within 10 years, in some shape or form, whether or not it's in the way that I've just talked about it or in another form, I think possibly it could be used. So, potentially at about the time that you guys will be leaving university and thinking about getting jobs in the science sphere, it will be happening. Could be happening. Yeah.
Wow, we've got about 5 minutes max remaining, if that's okay, Grace. So we're going to go through some more rapid fire questions now. And with apologies, we're at that regrettable stage where we're not going to be able to take all the questions that are
That's
coming
okay.
in.
I've probably talked for a little bit too long, but I just do love this topic.
Oh yeah, that comes across, Grace, really strongly. There was quite an interesting one, about the doctor who illegally used this on a human. The question is, wasn't the child born without the disease they were worried about? So technically, you could argue it worked. Any opinion on that?
Exactly. So the disease that they were was trying to cure was blindness. So there was a mutation that was causing a family to be blind, and so they had two babies that they took the gene out for blindness. And now, for privacy reasons and for legal reasons, we don't actually know what happened to those kids, because they're being protected because their fear of backlash against them in their association with essentially what was an illegal event. Yeah. So a bit of a shame because we won't know, but also understandable. Yeah.
This is amazing, and I'd be so fascinated even if I didn't have identical twin sons who are 4 years old next week. Are you worried? Last one on this theme. Are you worried the tech will eventually be sold with profit in mind and not ethics? So be very mainstream for designing babies rather than curing diseases.
Yeah, 100%, absolutely. There's always this industry versus science, and people are out to make money. But that drives innovation. So it's a really difficult question. I think yes, someone will make a fortune off CRISPR with some therapeutic one day. And it's up to the scientific community to keep making good quality research and provide competition to the field so that you don't get these monopolies. Yeah.
Can you recommend any good sort of super curricular resources, presumably quite accessible ones that you could follow up this fascinating talk with?
Yeah, that's a really good question.
And also, Grace, if you want to have a think about that, I can pop a couple of recommendations in the follow-up email tomorrow.
Yes, perfect.
If you want to
Let me
take
have a
time.
think about
that.
Great.
Because
it's been a while since I was at school, and I'm sure there's so much good e-learning stuff and platforms and stuff out there. So I'll ask around and I will find out what's been
brilliant.
used.
I won't be sending out that follow-up email until mid-afternoon tomorrow, so that would be brilliant, Grace. Thank you. That's a nice way of doing it, isn't it? There's a couple that I might be able to, certainly not the science content, but do you have to have studied chemistry A-level to be able to study this? Well, one way of approaching that would be, for example, the Oxford Biological Science course asks for A-level biology plus one of physics, maths and chemistry. So the straightforward answer to that is no, but it really depends on, different courses, different universities. Have a look at ucas.com, put in biology related course, and then you can see all the biology related courses that you can take. And it really depends on universities, depends on courses. How intense is an Oxford science degree? I'm considering chemistry and I'm nervous it will all be work and no time for other things. Can I make a suggestion? We have, and again these resources will be in the follow-up email, we actually have an ambassador chat platform where basically in a safe space you can just talk to somebody who's doing chemistry, biology and other degrees and ask them. They're not required to give the right answer. They shoot from the hip, completely frank. Just ask them. Grace, do you think an Oxford science degree, from what you see, is intense?
Yeah, I think particularly the chemistry degree here I have heard is very intense. But I think it's really probably one of the best quality degrees you could get in England. And so I think if working hard comes with its rewards. So if you're really academically minded and willing to put in the work and have a competitive application, then I'm sure you would get a lot out of it. But yeah, I don't know about the other ones, but chemistry I think is known as being really rigorous. Yeah.
And to pass on,
but apply to Oxford, there's so much more. Everyone, you will balance whatever you can out of Oxford or any university. Yeah, don't let that deter you. Yeah.
And to pass on what I've heard our students say in relation to that question, and of course you can ask them directly yourselves. Essentially, the answer is it's pretty hard work, and anybody who says, say, studying at Oxford, it's really easy, they're completely maligned, really, in that impression, to be quite honest. Yes, it's hard work is their response, but also they do have time to balance that off with non-academic enjoyment. The 8-week terms are pretty intense, they say, yes, but they're also riveting as well, and they don't just think about their subject for 8 weeks when they're in term time. They do other things as well. It's a cliche, but work hard, play hard, I suppose.
Yeah. I think if you love a subject, just go for it.
Yeah, Yeah, but as I say, do ask them yourselves, and I'll make sure that link to the chat is directly on the follow-up email that I send to you and others who registered tomorrow. Grace, thank you so much. You're just so passionate the way you've talked about this tonight, and
thank you. I could
really,
talk
really inspiring.
more hours, so I do love
and
it.
As I said to you when I first saw your presentation just beforehand, you know, it's just brilliant to see the references to women in STEM as well, and your own particular journey. It's absolutely delightful that you mentioned that, because generally other speakers haven't, but then they've been asked that question anyway. Oh, how did you get here, and what was your interest? And I thought it was beautiful the way you covered that as well.
Thank you.
We've got some just lovely thank you comments coming through. This is so amazing. Best webinar I've joined, and I've joined many. There's one comment. Thank you for the session. It's very interesting. Thank you so much. This has been so interesting.
Oh, cool.
That's a nice way to finish.
Thank
Thanks,
you so
Grace.
much. That means a lot. Thank you, everybody. I really am very grateful for that. Thanks.
And thank you to all participants for kindly joining us tonight and your absolutely brilliant questions, contributions to the polls. Yeah, brilliant. Join us in a couple of weeks for chemistry, if you'd like. Cheerio.
Cool. Thank you, everyone. Bye-bye.